Very good afternoon, all of you, and my sincere apologies for the delay because of some technical issues. So thank you for joining us. I would like to uh, give my regard and MERA-India team regard NMR to Professor Anuradha Chowdhary, who has accepted our request to be part of this series. So before we start, I'll just brief about Dr. Anuradha Chowdhary. Professor Anuradha Chowdhary is the Director Professor Medical Mycology and in charge for National Reference Laboratory Antimicrobial Resistance in Fungal Pathogens at Department of Microbiology, Patel Chest Institute, University of Delhi, India. She received her medical degree in 1992 and MD in Microbiology in 1996 from University of Delhi and PhD from Faculty of Medical Sciences, Redmond University, Netherlands, and had a postdoctoral fellowship from Mycotic Disease Branch, Center for Disease Control, Atlanta. Her research interest includes molecular ecology, population genetics, and pathogenic fungi, antifungal resistance mechanism, especially Candida and Aspergillus. The epidemiology of system mycosis also is one of her interests. She is currently working on the molecular epidemiology of Candida auris, drug-resistant dermatophytes, and as well resistant Aspergillus fumigatus. Her work on invasive fungal infections and has contributed widespread recognition of antifungal resistance in fungal pathogens, including the most significant pan-multi-drug resistance Candida auris and azole resistance Aspergillus fumigatus. She has driven the antifungal resistance agenda forward as a global public health threat. Her work has been widely cited and she has been highly cited researcher. Web of Science Clarivate 2021 list of 1% most cited scientists by citation for the field in the past 10 years. She has also been listed one of the world experts in mycosis by Expertscape PubMed-based algorithm in 2020. In 2019, she was conferred the Indian Research Excellence Citation Award in Medical Sciences. She is an elected fellow of American Academy of Microbiology, Indian Academy of Science, Royal College of Pathologists, Infectious Disease Society of America, European Confederation of Medical Mycology, International Society for International Chemotherapy, and many more. With more than 190 peer-reviewed publications and editorship in prestigious journals, which includes PLOS Pathogen, Journal of Clinical Microbiology. Professor Chowdhary is one of the most illustrious research scholars in the international review. So with this, her short introduction, I know I can, read many more pages. So I request Professor Anuradha Chowdhary to start today's talk. Professor Anuradha Chowdhary, all yours.
Thank you. Thank you, Dr. Sachin Sharma, for inviting me and actually giving this opportunity, this platform for promoting fungal infections. And fungal infections are one of the topic which are highly neglected and one of the most significant topic which I'm working for last 2 decades. And this platform will actually help us to promote the cause of fungal infections, which Dr. Rajendra Prasad can sincerely say because he's been working for last about 5 decades probably in antifungal resistance and Candida species. So thank you once again, and I am sorry to all the participants who are listening for the delay due to technical, function here, something. So thank you for your interest. And with this, I am going to talk about Candida auris, how it has emerged as a pathogen of global importance and how this pathogen has resulted in the worldwide recognition of fungi in the global world. So first few slides, basically I'm going to talk today about my journey with this pathogen, which we started in 2012-13 working, and now almost a decade, what all we deciphered here working in India. And also I'll give you about some updates about general fungal infections. So with this, I'll start my talk and my first slide. Okay. So just to tell you that this slide shows that how much fungal infections, what is the burden of the fungal infection worldwide. So if you look at this slide, you will see that about 1 million people, about 1 million people are affected by, they lose their eye by fungal keratitis. And this occurs mainly in the countries like India, where we have a lot of trauma and a lot of agriculture farm practices are there. And the most important thing is that about billion of people worldwide, globally, are affected by fungal infections. Fungal infection of hair, skin, and nail are the fourth most common disease on the planet after headache and dental caries. So with this, with global population affected by fungal infections, but these are the infections which are not systemically infecting the patients. So the patients who are infected systemically by invasive fungal diseases, they are about 1.7 million worldwide. And if you compare this figure with the tuberculosis or with malaria, so fungal top 10 fungal diseases, in fact, similar population, similar amount of people as by TB, and about 2 or 3 fold higher than malaria. So despite of these high fungal burden, we are neglecting our fungal infections. And we have witnessed during the time of COVID pandemic that how mucormycosis emerged as a serious threatening problem in not only in India, but in several parts of the other, several parts of other world. So with this, I will like to talk about the second slide. So these are the just two pictures, not to scare you, but just to let you know that how the spectrum of fungal infection varies. On one end of the spectrum are allergic diseases. So when I say allergic diseases, they are most common of the sinuses. A lot of people suffer with rhinitis, running nose, because of the lot of fungal sinusitis happening. And also this, not only it remains in the upper sinuses, but also allergic lung diseases. And to the other end of the spectrum are serious invasive diseases. And you can see on the left side of your slide that these invasive diseases can be one of the manifestation on the top, which shows the telleromyces infection, which is initially known as Penicillium magnifi, now known as telleromyces, and one of the AIDS-defining illness in Southeast Asia. And then you also see a lot of disfiguring chromoblastomycosis pictures here down in the panel, which just shows that fungal infections can be really devastating. And then you see a newborn with so many tubes. So the newborns, like premature newborns, are infected in the nursery, in the neonatal intensive care units with the Candida species, and the mortality of these infections are very, very high. So with this kind of varied spectrum of fungal infections, right from skin, hair, nail infections to the invasive infections, that is what fungal infections manifest worldwide. And next. So this is one of the important paper which I'll always share, because just to bring to your attention that when you look at the mortality data, so the top 4 fungal diseases are due to Candida, Aspergillus, Pneumocystis, and Cryptococcus. So if you look at Aspergillosis itself, which is a mold infection, it has a very high mortality. Generally, invasive fungal diseases have mortality up to 50%, despite the presence of antifungals, availability of antifungals. And in some cases, like in aspergillosis, if the diagnosis is not made in the initial 1 week, then the mortality can be as high as 90%. So with this kind of severe invasive fungal infections, if we have infrastructure of diagnosing these fungal infections, we can save millions of lives. And that is the agenda of Gafi or the LIFE, like these are the societies which are promoting fungal infections awareness. So if we can have the diagnostic laboratories of mycology where we can give the diagnosis early, we can save a lot of population. And with this, my next slide. With this, I come to Candida auris. So Candida auris, when it emerged initially in 2009 in Japan, just from the first case, the patient had an external ear infection, and the auris was identified from there and was named as auris because it came out from the external ear canal. And at that time, till 2019, this fungal infections had gone so high that this is the first fungal pathogen which has been listed by CDC in antibiotic resistance threat report as one of the global pathogen of urgent threat, which can cause urgent threat. Why? Because it causes invasive diseases, nosocomial hospital infections, candidemia, and it causes outbreaks of invasive candidiasis in patients who are hospitalized in intensive care units. It is transmissible from one patient to another patient through the hands of the clinicians. And on the top of it, it is one of the pathogens which is multi-drug resistant and sometimes pan-drug resistance. When I say multi-drug resistance and pan-drug resistance, it is important in fungal infections because we only have 4 classes of antifungal drugs to treat human infections, unlike bacteria, where you have hundreds of antibiotics. So if this pathogen becomes resistant to 2 classes of antifungals, then the developing world will not even have access to the other classes of antifungals to treat these patients. So because of all these factors, first time among the 4 bacterial pathogens which are listed as urgent threat, Candida auris is only fungal pathogen listed in this report. And also the CDC has put on the watch list the second agent which I work on, azole-resistant Aspergillus fumigatus. Can I have another slide? So just to give you a brief update of what Candida auris, as I explained that it was isolated from, it's an Ascomycetes yeast which was isolated from the external ear canal of a patient in a Japanese hospital. And in 2009, same year in Korea, there were 15 cases of Candida auris, isolates of Candida auris that also came from the external ear otitis media, external ear infections. That's it. But till that, it was not considered to be any yeast which is causing nosocomial infections or even the invasive infections. But then what happened in 2011, this paper came out from Korea, which published only three cases of neonatal fungemia, candidiasis, which was the first invasive Candida infection caused by Candida auris. And what was important in these 3 cases were that despite the administration of first line of drug fluconazole and amphotericin B, which is a cidal drug, despite of giving those drugs, the patients did not recover, and the isolate was resistant to fluconazole. So till 2011, the world has not given any recognition to this pathogen. So what happened in 2013? Like, can I have next slide? So what has happened now? I mean, this slide shows that what has happened from last 2009 till, can you click my slide? If you look in last 5 years, just go back. In last 5 years, about 50 countries have reported outbreak of Candida auris. From 2009 to 2011-12, only 4 or 5 countries were reporting Candida auris, and that too in a small number of cases. So in last 7 years, countries with widespread, all the 5 continents, everywhere Candida auris has emerged as a global pathogen. Next, and this is how the map looks like. And the map in the red areas depict countries which are showing the outbreaks, countries which are having invasive candidiasis, candidemia, and large number of outbreaks. So this is what has happened in the last 6 or 7 years. Can I have next slide? So how this story started? So the story started in our lab in 2013 when we were working and we were analyzing samples from 3 important labs. So first in Delhi, 2 important labs. And we identified a few of the isolates by commercial system, which were showing Himalayan, some other name, which was unusual for us. Then and these were the cases from medical ICUs, from surgical ICUs, from neonatal ICUs. And then what we did just simple ITS sequencing, and we blasted, and we found that the isolates were Candida auris. They were matching with the 3 fungemia isolates from South Korea. And not only that, next slide, just click. Not only that, we also saw that the susceptibility pattern, the fluconazole, the first line of drug which we use here in India, most commonly, most affordable drug, all the 98% of the isolates were showing very high MICs, were resistant to this class of antifungal. Not only that, the second class, voriconazole of the same azole, was also 50% of the isolates were also resistant to this. And we were surprised. And then next slide. And then we did a very simple AFLP technique, which was available to our lab that time. And what we saw that the isolates which we got from 2 labs in New Delhi and also from one of the lab in South India, 1,000 kilometer apart, they were quite similar. They appeared clonal. So the term clonal was coined because we were also surprised that these are the isolates which have come from facilities, from ICUs and units where the healthcare personnel were not common. So how this is showing very related clustering. So with this is happening in 2012 and 13. So what happened in next, please? At that time, with very, very limited facility available to us for the genome sequencing and for the interpretation, we took the 4 centers and we did less number of isolates genome sequencing. And what we found that 4 Indian hospital isolates were highly related. And if you see the percentage identity, 96, 99% of the isolates were similar. So it brought to our mind that these isolates were clonal and they have emerged in our country, and they have widespread disseminated. So, and the most important thing was that all the isolates are fluconazole resistant. Can I have next slide? Click, please. Yeah. So soon after that, within a year, we got a report of 2 cases of Candidemia from South Africa, which they wrote to our paper. I mean, they said, yes, Nilesh paper. I mean, says, yes, what Dr. Chowdhary is saying from India, we also have the similar 2 isolates of Candidemia, which were misidentified, which were there in the system. And we did the sequencing and find out that we had Candidemia cases. And the second outbreak occurred in Venezuela. Can you click, please? In Venezuela, where we had we had 18 cases of Candidemia. So till then, in 2016, so till then, this was the problem of the developing world. This was considered our problem, hospital infection problem, our hygiene. So this was not given any attention despite my promoting this in several conferences. And then what happened? How this bug started getting the international recognition from Western world? Because, next slide, please. Because this was the outbreak which occurred in London Cardiothoracic Center with 50 cases of Candidemia and probable colonization. So Silke, the microbiologist there, she contacted us and she said, oh, I'm like, how do we go? We are also experiencing this outbreak. So that time, the typing which we developed here, we used our isolates and their isolates. Can you have next slide? And then this was the kind of picture was emerged that UK isolates, although they were also clonal, but they were distinct from the Indian isolates and also the other isolates which are available from Venezuela as well as from Korea. So that was the time when this fungal pathogen got importance. And I'll show you in the next slide that despite of, can I have next slide? So this was the press coverage. Once this pathogen got the infection got into one of the ICUs in London Cardiothoracic Center, this was the news piece that drug-resistant, can you click? Drug-resistant Japanese fungus is spreading through British hospitals. And when they say Japanese fungus, Japan never had an invasive disease, so far also. So by that time, Japan had external ear infection, but they just promoted that this is a Japanese fungus which is spreading through the British hospital. And now I'm going to show you one of the clipping that how fungal infections are ignored and they're still labeled under bacteria. And this is not the problem in India or in the developing world. Please click. And you will see this is one of the clipping by the health journalist. And every time this journalist writes about Candida auris, it says bacteria, Candida auris bacteria. So this is the field of fungal infections. Despite working on this field, sometimes when I look at it, this is how the fungal infections are neglected. So can we have next slide? So then the CDC consortium was set up because Pakistan also was experiencing the outbreak. And that time we were approached, but because of all these issues of doing the testing with them. So what happened, CDC had this consortium and then they undertook this. And excellent work done by Sean Lockhart. they genotyped, they did the whole genome sequencing of isolates from East Asia, from South Asia, including the Venezuela, South Africa. And then they showed that the similar findings, like what we were saying, that Indian isolates are clonal, they said that the auris has emerged as a clonal clades in these continents, in East Asian clade, South Asian clade, and then also the African clade. So among these clades, there are thousands of SNP differences, but within the clade, within that geographic region, the isolates are very related, suggesting that they emerged independently in each of the continent, and from there the transmission has occurred. So this paper was a landmark paper, first time showing that isolates have different clades. And then we have a lot of data is now based from this study. And now we have a fifth clade from Iran, which is distinct from these four clades. So this suggested that there is a recent independent emergence in different places. Have next, please. So now I'll take you that to the lab part, So why was this auris hidden? Where was it? When it was in 2009, why were we not able to identify it? Because I tell you my experience. So here we did not have the Bruker, this Margitov system. In 2015, we got the system when we already had sequenced. And many laboratories will not do, diagnostic microbiology laboratories will not do sequencing. So the Bruker bio-based data system, we used our isolates to identify, we were not able to thoroughly say that this is auris, and we had to create our own database. Why? Because that time the database had only 3 strains, and the identification was difficult. So in this paper, we showed that the routine microbiology laboratories which use a commercial identification system like Vitec, which is based on biochemicals. So the biochemicals will always generate something. So they were generating auris as hemolani. So probably it was hidden under the cover, under the name of hemolani. And even if we use the sophisticated system like Margitov, you also have to upgrade the database. You also have to improve the protocols of extraction of the proteomics from here. So this was the paper which showed that please update the commercial identification database system. Next, please. So then a lot of people started working on this, and can you please click? And if you look at this slide, these are the important commercial identification system a routine microbiology lab will use. And if you see all these systems, they are still used, So they were misidentifying Candida auris as something. I mean, they did not say no identification. They say Hemolini, Rhodotorella, Famata, anything. So a lot of awareness were raised. And then finally, Vitec and other system, Microscan, they all have updated in last 3 years, 4 years, the system they have updated. They have included Candida auris in the database. Next slide. And still after updating the database, I'll show you that still till to date, we have some problems in identification. So then we also worked with a team, with bioMERA team for improving the protocol for the Margitov identification, because we have a Bruker database. And then this work, we worked with Vitec to improve the extraction procedure. Next, please. And this is a very interesting example. So if the auris was first time labeled in 2009 as auris, was it misidentified? Yes, probably it was misidentified before 2009. So this is one of the recent papers which came from France. We said that, can you click? So what happened that one of the patient in France, who visited India in 2007, and at that time, he was admitted here in Delhi in one of the ICUs and then shifted back to France. And there, this isolate was taken from the bloodstream infection. And then it was probably misidentified in their collection. So now with the updated systems, they sequenced it and found out that in 2007, before auris was labeled, so the isolate was C. auris and probably originated from India. So they did the whole genome sequencing and they found that the clade was quite similar to the Indian isolate. So here I like to tell you that when I joined Patel Chest, and we always kept our isolates. So now we have gone back to our system, and From one of the neonatal intensive care unit, we had one of the isolate which was probably mismatched as Candida glabrata at that time in 2007 as auris. So now we are working on that paper with, and trying to find out with this, how the correlation is with the 2007 isolate from France. Can I have a next slide? So even if now, I mean, this is a paper from London where they have evaluated different systems. And they, if you can see on the third line, if you can see that East Asian clade. So even the updated version will not yield identification of the East Asian clade of CRS, either with MALDI or with VITEC also. VITEC, it will give a misidentification. So laboratory personnel who are using, even if the updated database, and if they have a different clade of orders, there might be some issues for identification. So the purpose of this slide is to just spread that kind of information. Can I have next? So this is now the latest updated CDC website. So USA had all the clades. I mean, US got the infection, and then at present they have 806 reported cases of CRS and more than 3,000 cases of colonization. And if you look at the red spot areas, these are the cases where, these are the counties where more than 50 cases have been reported. And US, with all this migration and transportation, all the clade, South African, East Asian, all the clade isolates are there at present in US, and it is an important fungal pathogen. Although surveillance system in US are really good, I mean, they have now, it's a notifiable disease, 2019. So the data, what they are depicting here is a result of the surveillance cultures, colonization they pick up. This kind of effective surveillance is still not available in developing countries. So if I say the true burden, it is not available still in India. 1 or 2 centers which are working on auris. So similar happens with other part of the world. So now what I'm going to bring your attention to the resistance pattern of Candida auris. So this was in the same Lockhart paper, when all the isolates were collected, this data was given that 98% of the isolates are resistant to the first class, the azole drugs, fluconazole. More than 50% of the isolates, similar to like Indian isolates, are resistant to voriconazole. And also 1/3 of the isolates are resistant to amphotericin B. So here we have new data. I mean, these numbers are increasing. Amphotericin B, if I look at the 10-year data, previous 5 years and now, I mean, our resistance in amphotericin B, which is the most important drug used in India, not the liposomal, the conventional, because it is cheaper, and our physicians are still using it because, you know, they have experience, and we can't, lot of government setup cannot afford it. So the resistance of these is also going high. And echinocandin resistance is 7 to 10% at present here also. And if you look at it, 41% of the isolates are MDR. So, and 4%, which is quite a significant, and many places now this 4% is increased. So to which are showing pan-antifungal resistance. Can we have next? So, I mean, these slides, I'm going to primarily talk about the work, how we have done it. I am aware of a lot of work by Dr. Rajendra Prasad, excellent work on CDR1, on Candida auris, also the chitin contents, I'm aware, but these are the slides I'm going to show you about the work which we did here. So this was the first study which gave globally the importance of large number of isolates to show that what antifungal resistance, the burden of antifungal resistance is, and what kind of resistance mutations we were encountering. So in India, we did this study and we found that azole resistance was occurring. Probably we attributed that ERG11, which is a azole target gene. It had mutations, 2 types. But what was interesting was that some of these strains, about 5% of these strains were susceptible to fluconazole, and those strains were also showing mutations. This told us that probably ERG11 mutation is not the only factor contributing to the azole resistance. And also we showed that echinocandin resistance was happening, and this was due to mutations in the hotspot region of the echinocandin FKS1 gene. Can I have next slide? And this was the initial data of echinocandin resistance, which has been supplemented in the recent paper now. from India. But this was the paper which first time showed that echinocandin susceptibility is not the way it is done, because many misinterpretations of echinocandin susceptibility was happening. So this paper showed the importance of the mutations which rendered the isolate as resistant. Secondly, this paper also told, can you click please, that susceptibility testing will show of this class of drugs will show a paradoxical growth effect. When I say paradoxical growth effect, that means in the presence of high concentration of echinocandins, we'll have the MICs will be high, but that will not be correct. So we suggested that, look at the MIC at 24 hours and also look at the mica fungin rather than caspofungin. So this was one of the paper which shows the usage of susceptibility data. Can I have another slide, please? Another slide, please. So when I showed you that few of the isolates were susceptible but showing the resistance, then this paper by Reback, so they quoted that paper and started this story saying that even if when azole mutations were there in ERG11, there were isolates which were susceptible. So they worked on this tag1b marker, and they showed that decreased azole susceptibility was linked to mutation in transcription factor tag1b gene and also the overexpression of drug flux pump CDR1. So the similar, in one of our recent paper, we also showed and we also find out that the similar kind of tag1b mutations are present even in the Indian isolates. Can I have next, please? And then in addition to that, we also worked on the two-component response regulator, and we found that the signals transduction and MAP kinase signaling pathway, these both pathways are very, very important. It has been shown in Candida albicans as they are very important regulators of adherence and antifungal drug resistance and virulence. So when these two genes, one which is encoding the response regulator SSK1 and another MAP kinase HOG1, when both of these genes were deleted, then the susceptibility to two classes of antifungal, the amphotericin B as well as fluconazole, was increased. And probably this suggests that, these pathways, if we target and if we do more in-depth analysis of these pathways, this could offer a new pathway for the drug development. So this was one of the study which we did in last 2 years. Can we have next, please? And just to give you an overview, that I kept on saying about Japan did not have invasive infections. So this is one of the study from Christina Cuomo group where they did the genome sequencing and they studied the genetic arrangements, and they found that the clade 2 isolates, when I say clade 2 isolate, East Asian or Japanese isolates, they have a very highly rearranged chromosomal patterns, and their genomic stability is less, and there's a large subtelomeric regions which are deleted from these strains. So probably that is the reason which says that these isolates are susceptible. These isolates are easy to remove by disinfectants as compared to the isolates from the South Asian clades. So and also these isolates exhibited a different karyotype. So the genetics, the studies related to Candida auris genomes is still, they're just coming up because this pathogen is still very new to understand. Can we have next slide? So now then COVID. What happened in COVID? So because first, I like to introduce that this infection occurs mainly in ICUs. And how does it happen? When you have a compromised infection prevention control practices, when hand hygiene is not good, when gloves are not used properly. So this is transmitted from when the common areas and spaces are not cleaned effectively. So this fungus, it stays in the hospital environment. It persists in the inanimate sources, in the inanimate environment, and stays for a longer duration. From there, it can be transmitted to another patient. So when the COVID pandemic came, so we were aware that our hospital ICUs will be overwhelmed. So working in the field of Candida auris, we were prompted to write this commentary, which said that, the countries with high burden of C. auris should promptly identify and try to contain the outbreaks, especially in ICU patients where we will have lots of patients with mechanical ventilation, invasive devices. So soon after this, can we have next slide? early in the pandemic, from April to July, just 3 months in one of the ICUs, we worked closely. And we found that we screened about 596 COVID-19 patients who were critically ill, who were admitted in the intensive care units with several secondary bacterial infections. So there we saw that Candidemia was detected in 2.5% of cases early, very early. And then what we saw that C. auris was in 2/3 of the cases, Candida auris was the causative agent. And the most important thing was that the all-cause fatality was 60% in these patients. And Candidemia was developing in these patients after a prolonged stay in ICU after 10 days, from 10 to 42 days of admission. And also elderly people were more affected. And the most important finding was that patients with invasive devices, with mechanical ventilation, were having Candida auris. Probably they were colonized, and when these devices goes, then the invasive infection occurs. The most important part was, second slide, please, that majority of the isolates were resistant to several classes of antifungals. Similar, 70% of them were multi-drug resistant. So what choice is left? Echinocandins were left for treatment of these patients. And if you look at the yellow, all these strains have high, almost resistant to several classes of antifungals. Can I have next? In addition to Candida auris, there were also other species, but they were few. But if you look at the profile, they were not as resistant as Candida auris was. Can you have next? And then soon after that, see, this was the first outbreak after we reported this from Florida. And CDC intervened in this. CDC helped them in understanding the transmission dynamics. The Florida healthcare facility in July 2020 reported that they had 3 cases of Candida auris bloodstream infections in COVID-19 patients. And they said, and then the CDC team investigated the prevention compromise practices, what were happening. And they realized that, prolonged usage of PPE, and healthcare personnel were using double layer of PPEs and not changing the inner layer. So that was also getting colonized while using that. Also, the common shared equipment like computers, keyboards, all of them were positive. So when the emphasis was given for better hand hygiene, for effective PPE, then they were able to transmit, they were able to block this transmission from one patient to another. So next slide. So then what was missing with us in the first study, because it was early in the pandemic, that we didn't know whether this auris was already there, or of course it was already there, but we didn't know whether the Candidemia incidence has increased per se in COVID ICUs or not. So what we did, we selected 2 important ICUs, one in North India here in Delhi and one in Jaipur. And then we did a case control study. 1:2 case control matching was done. And we compared the incidence of Candidemia during the time of COVID and 1 year previously. And we observed that we recruited about 2,300 cases of patients who were admitted in ICUs. And what we saw that 1.5 to 2-fold increase in Candidemia occurred in the ICUs in 2020 and 21 as compared to 2019. And what we also observed that Candida auris was also again predominant in both the facilities. Can I have next? And when we did this, what were the factors responsible predicting Candidemia? We saw that there were 3 very important factors. Can you click, please? One was the prolonged ICU stay. Can you click, please? One was these were the multivariable regression analysis. We saw that use of anti-IL-6 inhibitor, tocilizumab, was associated with was one of the predictor for the development of Candidemia. And we also saw that, which we didn't see, before that, not many studies have shown that IL-6, anti-IL-6 is one of the important parameter. So we also again showed that duration of ICU stay was related to Candidemia, 24 versus 14 days in patients who did not develop Candidemia. And also severity parameter, one of the parameter was ferritin, was found high in these patients. So this was one of the case control study which added the importance of Candida auris Candidemia in ICUs, where we documented that the incidence of Candidemia is increasing. Can I have next slide? So if you look at this paper, which was published in CID regarding anti-IL-6 inhibitor, so it also says that, they also observed a lot of bacterial pneumonias Staphylococcus aureus and all in patients in which this anti-IL-6 was used. So this is how the story evolved. Can I have next, please? And then just to give you one of the example, that this is a study from Spain, and what they observed that during the time of pandemic, Candida auris, which was negligible before in 2019, 40% of the cases were due to Candida albicans. But once these ICUs had COVID ICUs, were turned into COVID ICUs, Candida auris counted for about 40% of the infections, and majority of the cases were replaced of other Candida cases were replaced by auris candidemia. And this correlated with the extensive usage of antifungal consumption in these hospitals in Malaysia. So probably antifungal usage was linked to emergence of Candida auris. Can I have next slide? And now coming back to my own study. So in this study, not only we did auris, but also it was interesting to see we did MLST for Candida tropicalis, because that was the second most important pathogen in the ICUs, in both the ICUs. Even Candida tropicalis was resistant to fluconazole. And both the ICUs had similar MLST type, similar genotype, which was transmitted among the patients. And even the resistant genotype was transmitted from one patient to another. So this was what we observed, not only related to auris, but also related to tropicalis. Click, please. And this tells us that this is not even India. I mean, everywhere in the world, resistance and resistant isolate transmitted from one patient to another is happening. And this was the study from Italy, where the patient developed Candida glabrata pan-echinocandin resistance while on echinocandin therapy. And that was a fatal case. So this slide just shows you the importance of resistant isolates, and the outcome of these patients will be fatal if we do not take into cognizance about the importance of antifungal resistance happening here. Can I have next slide? So now just to bring to your notice that whatever happens in US is always given attention. So this is one of the pan-echinocandin resistant isolates. 3 pan-echinocandin, pan-resistant isolates were observed in New York facilities. And from 2016 to 2019, a total of 801 patients were screened. And they observed that 3 patients had pan-resistant phenotype, although these patients had never traveled outside US. Can I have next slide? And when you look at the factors of these patients, one of the common thing was the usage of the patients are ventilator dependent. So, and also echinocandins were used in these patients. So these were patients with underlying diseases. These were the patients with mechanical devices, but they did extensive sampling of the environment, and they were not able to identify resistant isolates in the environment. So can I have next slide? So just to give you a small brief about that, how Candida auris in the early 2006 till 2017, where US was just analyzing the cases and they were experiencing these outbreaks, how this pathogen has spread to so many hospitals. So what they observed that long-term care facilities, which use ventilators commonly. So patients who were admitted in long-term care facilities were transferred to another acute care hospitals or to full hospital tertiary care centers. So from there to another hospital, they were never screened. So once these patients go to another facility, how they transmitted it to several patients. So if you click here and you'll see the connection between one patient. Can you click, please? Yeah. So if you look at these boxes, these represent the hospital. And if you see the solid lines, patients transmitted from one hospital to another, or one acute care facility to another hospital, and how these patients were carrying this pathogen, and how this was transmitted from one patient to another. So this was happening when effective surveillance was not there. Of course, now US has a very effective surveillance system, and they cohort the patients, they screen the patients, but this was a time in the initial time when auris was just emerging. And if you click the slide, and you'll see this, the connection. You could please click. So if you see these red, blue lines, they just show it. I mean, to me, it appears like a London tubes, one train from another. So this is the connection between one hospital to another. So this is called web of C. auris, how it is transmitted. Can I have another? So now, during the time of pandemic, we also got enough time to study the chronic respiratory disease patients, and we wanted to see how the transmission is happening in our settings. So we screened all our patients of chronic respiratory diseases. Why we took chronic respiratory diseases? Because they are admitted one after few months. They are the patients with asthma, with COPD, so they really need hospitalization. So we screened all of them at the time of entrance, and then repeatedly, one weekly. So what we found that 37.5% of patients, they developed colonization by C. auris just after 1 week of hospitalization. And we were also screening the immediate fomites. We were screening the surfaces, we were screening the floor, and what we found that 10% of the fomite samples became positive within the week of the patient becoming positive, because C. auris is shed from the skin scales and it colonizes, it is there in the environment, it remains viable in the environment. And then we also observed that 25% of patients at the time of admission already were positive for C. auris. And why? Because they were repeatedly admitted and out and in patients. So we did whole genome sequencing, and we found that multiple genotypes of Candida auris colonize the axilla, groin, ear, nose. And one genotype is distinct from the other genotype, but in the environment, we found the same distinct genotypes. It's something picture very similar to other fungal pathogens where multiple genotypes are found in the environment and infection occurs with selective. And here we also, unlike the New York outbreak of resistant isolates, we were able to find isolates in the environment which were resistant to amphotericin B. So this is just one of the preliminary study, but it sheds a light, that how this is transmitted in our settings. Can I have another slide? Now, during 2020, that last 2 years have been a really fruitful journey for us. And this was the hypothesis given by two very important scientists, Arthur Casadevall and Dimitrios Kontianis, who are actually working on global warming. And they suggested that, still this hypothesis, no Candida auris was, not even a single strain was isolated from any of the natural environment. So it was always considered to be a pathogen of hospital, probably developed resistance in the hospital because of overusage of fluconazole. And they hypothesized, and one of the feature of Candida auris is it grows at high hypersaline in the hypersaline environment in 10% sodium chloride, and also it grows at 42 degree. So both these features, probably those features are related, and I mean, according to this hypothesis, they said that probably because of the salinity, Candida auris is probably present in the environment in the wetlands because of the saline preference. And because of the global warming, this pathogen has adapted itself to grow at 42 degree. And from the natural environment, probably in the natural environment, it existed as a plant saprotroph, like other fungal pathogens. Then how it is transmitted to humans? It's the hypothesis was that probably the avian host, which can, like Cryptococcus, can grow at, they have high temperature. So probably auris retained in their body, and from there, it migrated to the urban settings. And from the urban settings, from patient to patient, from humans to humans, from humans to hospital, it went to the clinics. So this was the hypothesis based on the high temperature, based on the salinity. So when we read this hypothesis at the time of the COVID, and then we had a, I was very inquisitive. I've been working in auris for a long time. So we contacted the Ocean Marine Department in Andaman Islands. And why? Because Andaman isolates had two kind of, they have all tidal marshes, they have swamps, they have hypersaline environment, plus they have areas where humans are it's not accessible to humans. So restricted entries to people from outside. So, we marked, I traveled and we marked certain areas that we wanted to see. From there, we wanted to collect. So we contacted and collaborated with them. Can I have next slide, please? And then We did Andaman Islands. We had 10 sites which we selected, and we collected seawater. We collected tidal marsh seawater. From beaches, we collected the water, and then we analyzed the samples here. To our surprise, we got Candida auris from two important locations. Can you click, please? And these two important locations I'll show you. So one was the tidal marsh where there was no human activity, and one was the beach where migration was allowed and people were there on that site. So both the sites we collected about 24 CRS strains. So what was the difference in these two sites? Can you please click? When we did the susceptibility testing, we found that the isolate, which we got it from the tidal marsh with restricted human activity, was susceptible to fluconazole. And if you click, please. And then when we did the temperature analysis, we found that this isolate was statistically significantly showing slower growth at 37 as well at 42. It was trying to adapt itself. So that probably suggested us, this was the first study which showed that C. auris existed in natural environment. C. auris was probably susceptible when it was in natural ecological niche. C. auris probably developed resistance later on and acclimatized to global, increase in the temperature. But this was and also the isolates which we got from the beach, they were quite similar to the human isolates, which we did last, like 50 isolate genome sequencing have been done from our center, and we correlated with that. So those isolates were quite related, but this isolate was not. So this all suggested that probably environment is the source of C. auris. Can you click, please? So this was considered to be a very important landmark study, and the same authors, Kasadaval, had written in one of the commentary for this paper that this is a landmark discovery, which is very crucial for understanding the epidemiology, ecology, and emergence of C. auris as a human pathogen. And this was also covered by ASM press release. Can you click, please? And then can you again click, please? Yeah. And now I'm going to share something very, very interesting, and which is still not out. And this is one of my last 2 years' effort. The first study prompted me to do the work that how come these strains are developing resistance in the environment. When we have susceptible isolates in the coastal wetlands, how come this resistance is developing? So for last 2 years, we've been working on lots of fruits. And we found out, I cannot share much because it's still it will be released next week, and there is a lot of coverage of this paper. So but I still will like to just tell you that now we can say that drug resistance is probably attributed to the usage of antifungal drugs in the environment. Here I'm going to tell you that like Aspergillus fumigatus, azoles class of antifungals are used for so many years everywhere in the world, which drugs to prevent the infections of lot of for fruits, for vegetables, fungal infections which destroy these cultivars. So against this, we are using fungicides, and these fungicides has a similar structure what we use in medical azoles. And we found that stored apples were harboring Candida auris. They had resistant phenotype. And when we did susceptibility to the azole fungicides and compared those azole fungicide strains with the Andaman strains where there was no agriculture fungicides were used, tenfold higher resistance was observed against these fungicides on these strains. This is the first study which probably hints that the resistance development which is occurring in Candida auris is probably related to the usage of fungicides in the environment. And also there are certain can you click, please? Certain important points which also shows that these strains are very unique, and also these are strains which are related to patients. So what is happening in apples, you will read the paper, and we have also done a lot of pesticide detection in this, and it's a good collaborative study, and hopefully this will open avenues to a lot of other researchers who are working in this field. Next, please. And here I like to emphasize, this was the commentary which I had written for azole-resistant Aspergillus fumigatus, and I seriously believe everything is in our environment, and the concept of One Health really needs a lot of emphasis. I mean, our health is related to our environment, is related to our to animal health. So everything is so connected. So with this, I mean, can you give me next slide? I will just conclude by saying that Candida auris is an important outbreak pathogen. Several important researchers are working on this field, including Dr. Rajendra Prasad. And this is one of the pathogen, not India-centric. I mean, it is now a global pathogen. And it is a major infection control challenges. It creates a major infection control challenge. But because of this, we have gained people who are mycologists, people who are working on fungal infection. We have gained, can you have a next slide, please? Lot of importance. Why? Because GLASS, Global Antimicrobial Resistance Surveillance System, till now, for last 5 years, has been just covering resistance of bacteria. So this is now, this year, this is the first time that GLASS has included Candida from bloodstream infections, compulsory to be included in the GLASS surveillance program. Initially, we are just going through the stage of pilot protocol. So one of the pilot protocol was developed when I was working with them. And very happy that now we will actually have a true burden of resistant Candida species worldwide. At least we will know what is happening in globally, because WHO has now given us a platform to submit the data. Can we have next slide, please? And can you click? Although this was in 2006 when I was visiting CDC, Dr. Warnock has written this. And I truly believe that fungal diseases, it holds true today only. Fungal diseases are evolving public health challenge. And we will keep on seeing the emergence of new fungal pathogen, and we will also see resurgence of mycotic diseases, what we saw, mucormycosis, during the time of COVID pandemic. And these are not uncommon. They are growing public health problems, and a lot of awareness has to be raised, and a lot of hard work has to go for fungal infections, their detection, promoting the infrastructure, right diagnosis, more antifungal drugs. And that's the only agenda which I work for. Thank you so much. And next is my last slide, please. And this is the most dynamic team of past and present of Candida auris. I mean, it just started with very small PhD work, just dedicated patient care. That's from there it started. And now the lab is a national reference lab for detection of antimicrobial resistance in fungal pathogen. And now we are working with 30 states. So this is how it is. And thank you very much for giving this opportunity once again to promote fungal infections. That's it. Thank you.
Thank you very much, Professor Anuradha, for this wonderful talk. So now, I request people can write their question in the chat box, and we can pick some of the questions from there, and we can ask on their behalf. Meantime, I can request Professor Rajendra Prasad. I'm sure he has some questions for sure.
My privilege, actually. Dangerously fascinating, Anuradha.
Thank you, sir.
Really, and the kind of dedication continuously you are working on this, really it has given real good insight what's yours and what it can become even after so much work. Really. It's very dangerous, very dangerous.
Yes, sir.
I had just a small question related to COVID infection and then kind of auris, which you detected in patients. So how does it compete out with the mucorals, the black fungus?
How does it compete? Sir, there are two different scenarios. I'm talking about the intensive care units, sir, where the patients are mechanically ventilated or they have several invasive devices. Although their blood sugar is taken care of, right? So if you see that this is hospital acquired, but when mucor happened in the second wave, and at the second wave we had, doctors were just seeing patients, prescribing steroids, and not taking care because it was not possible. Hospitalized, these were the not patients which were hospitalized. There were majority of patients who were coming from home with blood sugar not managed properly. So that kind of underlying disease were different. They were patients with who were diabetic, and those who were not even diabetic, but they were given steroids, they were borderline. So that was another scenario. So if you compare, one cannot compare the
No,
mucormycosis risk
I
factors.
didn't mean to compare. What I meant was, did you also see the co-infection of both?
Yes, sir. Yes, sir.
That's
I
right.
mean, the first time when I saw, I was surprised in the sinus, we got the fest tissue. So in the sinus, there was auris and there was mucor. So because it can be in the nose, you know that it can be colonizer. So first time I didn't pay attention. But when the biopsy histopathology report came later, so then we saw that in deep also, Candida auris was also there, deep tissue infection and the mucor. Because mere isolation will not say that it was causing infection. So yes, I saw. And one or two case reports also I saw later of auris and mucor. I think it was in medical mycology, but I was like so much occupied. I thought, yeah, I know it's going to happen, but didn't report that. Yeah, it was.
So when you say this recent paper, which is going to be, of course, very hot,
Yes.
that is acquired resistance.
Yes, sir.
So what about these? In case of bacterial resistance, we have these ancient drug resistant genes, which
Yes.
people have discovered 30,000-40,000 years earlier from the permafrost deposits. So the genes always existed in nature, and is that they are adapted to become to have this kind of role.
I completely agree, sir. Completely. I mean, I can't disclose here, but those tag 1B, everything we did in this paper. Because just because it is under the coverage for press release. So, I mean, I'm not, but yes, what you're saying is absolutely correct. The emphasis here is the fruits had pesticides, a lot of fungicides, tebuconazole, bromiconazole, similar to what, and that is not happening in India. It's everywhere. You pick up anywhere because of storage. Everyone uses, right? And then these strains had a very high MIC to azole fungicides from here as compared to the strains which are from the baseline, from Andaman, where there was no agriculture land. So one hypothesis. Second, when you read, you'll find a lot of interesting thing like wax. Wax, when you wax the artificial wax, it has a lot of fungicides you incorporate, which stays there. So that also we did in this paper. So yeah.
So I stayed on Sachin first because of Anuradha, and second, I also support that fungal infection should not be neglected.
Yes, sir.
And,
Thank you.
you guys should really consider us. We are also important.
Yes. I mean, I was
Thank you.
surprised to get because it's malaria I had. And I thought, okay,
Yeah,
fungal
exactly.
infection, no one will be interested. I mean,
Right.
why? But I thought whatever opportunity I get, I promote fungal infections.
I
And I
agree.
was so happy to see you also in this forum. Thank you for joining and supporting
Nice.
me.
Thank you, madam. So as this the name of the series is infectious disease series. So nothing to do with particularly with malaria, but I can see a common interest, which is climate change.
Yes.
See very clearly on the both sides. So I was
Yeah.
just wondering. I was listening your talk. Did you get any chance uh, to have a comparative analysis, the isolates you got in the COVID times or before the pandemic, or I don't know if you have contacted these people outside in Florida. Can you check the geographical location based some changes, genetic diversity between these isolates? That I think that would be interesting to see.
Yeah, it would be, I'm sure, because auris grows at 42. So it's a universal fact. I mean, any strain of auris, whether in US or in India. So, I mean, I can't say it. Good point, but no, we haven't checked that.
Okay, this is also interesting that we found this on apples, and based on this, we have this question: did you check the Candida auris in other fruits and vegetables?
Yes, yes. I mean, we did 96 different, 9 species of fruits, and tropical and temperate, and also freshly plucked, because we wanted to say why we are getting it stored.
Yeah.
And the freshly plucked from organic and non-organic. And there was a huge difference. So we waited for 1 year for the apple harvesting season in India, and we went to these orchards and plucked those apples. So no, they did not even have a much yeast diversity as compared to the stored apples. So, and this is not here. I mean, a lot of studies have done in the past which have shown that fruits can be a transmission or reservoir of transmission of drug-resistant strains, but never auris, I mean, no, never. Before that, from a study from China has shown that tropicalis can be there, because these are more human pathogens. They are accidentally there, right? I mean, they're not, ecology is there, is specific. But that's why, to prove that point, we also did conventional versus, but it's a very small study in the sense that we had 20 from the freshly plugged areas, and then 60 from the stored, but different time frame. So this is just a study which gives you an insight about existence. Maybe a lot of other people will now start working and they will find other fruits. I mean, sure, why not? Wherever we are using antifungals in the environment, that can promote the drug resistance. So yes, we did a lot of other fruits also, temperate, tropical, but because probably because of the artificial waxing, and auris is isolated from the external ear canal with a wax component in ear. I mean, all these are hypotheses. We have tried to do some experiments, but I mean, it's just a small study which says, gives a direction.
Yeah. Okay, so I think Sonal and Bhavna have some questions. Sonal, you can ask.
Thank you, sir. Ma'am, I have one question. Have you seen any case of co-infection of Candida auris with malaria parasite, especially in case of severe malaria cases with Plasmodium falciparum?
Not here. That's a lacune of only mycology lab. No, but very interesting. I mean, now I'll keep it. No, I haven't.
Oh, okay.
I haven't received, I mean, I don't see those cases. So that is why. Probably it would have been there, but we never received those samples.
Okay. Thank you, ma'am.
Very interesting question. Stimulates my
Bhavna.
mind to do.
Ma'am, the finding of auris. Ma'am, your finding of auris in uninhabited islands of Andaman is a really landmark finding. Now, the biosphere is always in a churn, and from microbes to microplastics or carcinogens, toxic chemicals, they always end up in places where far from where they have originated. So how do we, address this egg and chicken dilemma? Like, how can we be very sure about
Just because that is what we wrote, that the study says that auris can survive in the natural environment. How it is transmitted to humans and vice versa still need to be deciphered. So you are absolutely right. The only thing which I gave is that certain strains from beach where humans were there. So there, these strains were similar to the human strains, specifically the Indian clade 1 strains. But how, whether it was in the patient which was from the in the beach, it was from the patients who were shedding and it reached there, or whether it came from the plant saprotrophs, and because there were vegetations in these places. So whether it came from there is still a mystery. I mean, don't know. You're absolutely right. So that is why this study says that yes, C. auris can be detected in natural environment. It persists in natural environment and with a different phenotype as compared to the health settings. So who came, how came, is a long journey. It just happened last year, and then now we have another paper for ecology of C. auris. No,
Right. Thank you,
This
ma'am.
need to be worked.
Right, ma'am. Ma'am, also, auris is so highly prevalent in India and all the hospitals. So, is it possible that it's transmitted from the hospitals and it has moved to the uninhabited places? It's spreading and saturating the other places now.
See, the how it is transmitted is because it colonizes the human skin, and once it colonizes the human skin, the skin sheds, right? So
Yes,
that when it
yes.
sheds, so it persists in the inanimate environment down. So of course, with the colonized healthy humans, it can be, but it doesn't infect those people. It doesn't infect the normal humans, right? So when these patients are admitted with several diseases, underlying risk factors, when the skin is breached, when the immunity is low, when there are major invasive devices, then only it infects. So, normal human beings who are admitted in hospital will have a colonization, will come back, can shed. But this doesn't mean that it's community acquired or it's doing some infection. It is only infecting people who have underlying diseases, who are in medical ICUs, intensive care units.
Thank you so much, ma'am. It was a wonderful talk.
Thank you so much.
Okay, we have a sort of question and suggestion. As the resistance is coming from the environment, we should search for the environmental solutions like AMPs from insects can prove to be effective.
Yes.
So what's your suggestion on this?
So I always say that a lot of other practices, biopesticides, I mean, we cannot say stop this. That is not a solution, because more than billions of people have to get their food by agriculture. These things have to be used in the environment. Otherwise, we'll not have that crop yield. But switching over slowly to more non-medical antifungals is the solution. Because if the same drug is used in humans and the same drug is used in environment, it's not only this pathogen, slowly we'll also have lots of other resistance emerging. So concept of biopesticide, concept of other farming has to be really strengthened. And also why we're using multiple antifungal fungicides, so that also has to be slowly seen, and practices has to be changed. And all this will come when we all sit together. I mean, environmentalist comes out with another solution. So at present, I mean, I can't say that stop this, but I can say that there are certain ways where you can have other effective ways of using these compounds, much healthier compounds which do not interfere in human health, which should be promoted for these kind of farming.
There's another question: which form of Candida shows more resistance, hyphae or spores?
See, this one, Candida auris, does not have, rarely it forms hyphae. So this is always a blastoconidia. So when blastoconidia invades, it gets elongated, right? So it is one of the form. Blastoconidia is there in the patient's skin or anywhere else, and when it invades, the cells get elongated for Candida auris. For Candida albicans, it is a pseudohyphae. Once the pseudohyphae is formed, then they form, then they invade. They are more invasive. So, but auris is not like that. Auris has a blastoconidia. What you call spore is a correct term, is a blastoconidia.
Okay. So with this, I think can we conclude, Bhavna? If there's no more questions, yeah. Okay. So with this, I would like to thanks to all the participants, all the people who have joined us, Dr. Rajendra Prasad and other colleagues. Thank you. My very special thanks to Professor Anuradha Chowdhary. She accepted our request for this, and I'm really, glad to see that you have, openly discussed your work. In fact, what work is in progress, you have showed, gave a little taste of that also. So with this, thank you very much for joining us. And because now situation is better, I request you please visit us at NIMR, and we can see if any collaboration is possible between us.
Absolutely. Look forward. And that's how new science gets generated. Thank
Right.
you so much.
Thank you
And
so
I'm
much.
aware such a great work NIMR is doing. And I am so happy to see, the platforms which you have developed for this. And I've been in touch with a lot of series. I was watching yesterday also the last one. I mean, amazing work your team is doing, Dr. Sachin.
Thank you.
Really delighted to see. Thank you so much. And
Thank you
huge
so much.
congratulations to all of you.
Thank you, ma'am. Thank you so much. Okay, see you. Bye-bye.